To get started, running a little late. Hope everybody had a great break. We have this one last lecture before Wednesday's exam, and what we talk about today is fair game for Wednesday's exam. And don't forget that'll be at 5 o'clock on Wednesday afternoon. And so where we're headed now is we're going to talk a little bit about the brush border enzymes. We'd finished last Wednesday talking about the brush border enzymes that were dealing with the disaccharides, those so-called disaccaridases. We emphasized the concept of the unstirred layer. We emphasized the idea of the sodium-driven co-transporters. Well, we're going to finish that story here with the idea of brush border peptidases. Just as there were transmembrane proteins in the form of disaccaridases in these enterocytes, the apical membranes. In this case now, they're going to be carboxy and aminopeptidases. Now you remember in the bulk phase, well, the digestion by trypsin and chymotrypsin had been to take large proteins or large peptides and convert them down into much smaller peptides, but still peptides. Those peptides are too large to be absorbed by the enterocytes, and so the final stage of digestion of proteins will be where the amino and carboxypeptidases clip off one amino acid at a time from the either amino or carboxy termini of those proteins, and then those individual amino acids are transported by a sodium-driven GOAT transporter into the enterocyte. So we'll start there. And then what we're going to think about is what the fate of that glucose and the glucose and the galactose and the fructose were from the disaccaridases on Wednesday. And now we're going to think about, we're going to add in these amino acids and think about where they go. Because once these digestion is finished on the brush border, and those single unit amino acids or hexose sugars are transported into the enterocyte, they're now going to come out the basolateral side. But they're going to come out of the basolateral side, and they will be strategically located right next door to capillaries of the hepatic portal vein. The capillaries of the hepatic portal vein are going to gather these amino acids and glucose molecules and other hexose sugars, and they're going to then transport them to the liver, because that's where all this stuff is going. And it's going to the liver through something known as the hepatic portal vein, or the hepatic portal blood. Now, the hepatic portal blood will contain the glucose and amino acids from the enterocytes. That blood then is going to go to the liver. And we have to talk a little bit about liver structure to understand what's going on here. We'll see that the structural and functional unit of the liver is something known as the liver lobule. And the liver lobule is unusual in that it's hexagonally shaped. And at each vertex on the hexagon, there will be 3 tubes. One tube will be for the hepatic portal blood. This is going to be nutrient-rich and oxygen-poor blood that's come through the intestines. Then there's going to be also a mixture of blood in the liver, a type of blood called the hepatic artery blood, and that's going to come right off of the aorta, so it's oxygen-rich. And then the third tube is going to be the bile duct. Now we're going to see that the hepatic portal blood and the hepatic artery blood will mix together. They mix together in a structure known as the sinusoid. Now the sinusoid is just a specialized term. The sinusoid is nothing more than a capillary. We've seen capillaries over and over and over again. And the capillary of the liver is called a sinusoid. So within the sinusoid, we're going to be mixing this nutrient-rich, oxygen-poor blood from the hepatic portal system with oxygen-rich blood from the hepatic artery. The ratio is about 70%, 75% hepatic portal blood, 25 to 30% hepatic artery blood. But we do need an oxygen-rich source of blood for the liver cells. Then we mix this blood in the sinusoids, and then we'll see that the blood particularly carrying the nutrient-rich blood from the hepatic portal vein, that blood now is going to contain large amounts of glucose and amino acids, and those amino acids are going to be transported into the hepatocytes. Once they're in the hepatocytes, then they're going to be transformed, because we'll see the function of the hepatocytes is to convert glucose to glycogen. Remember, we mentioned briefly what glycogen was. It's a kind of animal starch. So it's this polymer of glucose. Usually it's highly branched. There's lots of 1,6 connections. So the hepatocytes are then now going to convert the glucose to glycogen, but they're also going to be converting amino acids to protein. So the whole story here has been to take large molecules, break them down into the component parts, and now in the liver we're going to reconstruct them. The other thing that goes on in the liver with the hepatocytes is something known as bile synthesis. Now, we introduced bile some time ago as a waste product of the breakdown of the porphyrin rings. Well, this is a different kind of bile. This is a different molecule altogether. When we talk about bile salts and bile acids, we're talking about a modification of cholesterol. And we're going to take cholesterol, and we're going to take that cholesterol molecule, we're going to add at one end to it either a molecule of taurine, which is charged, or a molecule of glycine. Now we have a charged end to this cholesterol molecule. We have a charged end to the cholesterol molecule, we have an uncharged region of the cholesterol, and so now we have this amphipathic molecule, and it's basically a detergent. And what it's going to do is it's going to break these large fat droplets or fat globules that you've taken in with a big meal, a big fatty meal, and break it down into tiny droplets. The reason we have to do that is that the lipase, remember, which was secreted by the acinar cells of the exocrine pancreas, the lipase can only work in an aqueous environment. So it has to work on these tiny droplets rather than the big globules. If you imagine this great beach ball of fat and you have to work at it from the edges in an aqueous environment, then it's going to take forever to digest it. So with emulsification, we take this beach ball and make it into BBs. Those BBs now have a much more favorable surface to volume ratio, and we can now digest the lipid in the BBs with the lipase. But it doesn't end there. We're going to use this, we're going to digest these triglycerides into monoglycerides and fatty acids, and that's going to form a micelle. Remember, early on we talked about micelles, and we said we'd re-enter this idea of micelles late in the course, and here we are. We're going to make micelles here. Micelles are stabilized by the bile. And the micelles, though, because they contain monoglycerides and fatty acids, once those micelles enter the unstirred layer, then the monoglycerides and the fatty acids can go right through that hydrophobic phospholipid bilayer. So they go right into the cell. And then a bizarre thing happens. We take these monoglycerides and fatty acids and we now reconstruct them into triglycerides. We do that in the endoplasmic reticulum. And then once these lipids are moved from the endoplasmic reticulum to the Golgi apparatus, in the Golgi apparatus we complex them with proteins. That gives them a little bit more charge so they can interact with water a little bit better. Then we package them into something called chylomicrons. And the chylomicrons then are packaged into vesicles and are secreted from the basolateral side of the anthracite. Those lipids now don't go into blood, though. They go into the lymphatic system. They go into this special structure called the lacteal. So they'll go into the lymph. And in fact, these chylomicrons are so big that if you take a sample of blood right after you've had a big meal, an hour or so after a big meal, and you spin it down and look at the plasma is cloudy. It's cloudy because the blood plasma has received the lymph from lymphatic systems into the subclavian vein. And those large particles of the chylomicrons are so big that they're actually reflecting light. And so you actually see that as a cloudiness in the plasma. And then if there's time, we'll finish with this idea of the endocrine pancreas. We've emphasized the exocrine pancreas. There's another aspect of the endocrine pancreas we touched on when we talked about the Russian experiments on dogs. This is where we make insulin. This region of the endocrine pancreas where we make insulin called beta cells. The beta cells recognize when there are high levels of blood glucose and they secrete the hormone insulin that causes muscle, both heart muscle and skeletal muscle, to take up glucose. But in order to do this, we need to have two players. We need to have the beta cells release insulin. The insulin now has to bind to receptors on muscle cells. And then the insulin binding to those receptors causes the movement of another glucose transporter to the membrane surface called GLUT4. That allows glucose to come in. One of the reasons I want to point this out is that diabetes is a very common ailment in our society. And there are two forms of diabetes called type 1 and type 2. Type 1 diabetes occurs when the beta cells are destroyed, usually by an autoimmune response. And when those beta cells are destroyed, we can't make insulin anymore. But the second, more common form of diabetes is called type 2 diabetes. And type 2 diabetes results from the lack of receptors. We don't know why, but for some reason we lack sufficient numbers of receptors to bind to the insulin to put enough GLUT4 on the surface to allow glucose to leave the blood vascular compartment and go into muscle. And so again, we see over and over again in here that it's the problem of the receptor, and that's the most common form of diabetes, is the lack of a receptor. But let's start here. Let me show you a slide, because I think this is an important slide. You need to sort of have this idea in your head. This is an image of a villus. Now remember, the villus is a multicellular structure. So the villus is constructed from the enterocytes. So all these little hash marks here, these are all enterocytes lined up side by side. And this finger-like projection is the villus. Now we're going to draw in a moment the finger-like projections of each individual enterocytes that form each individual enterocyte, and that forms the brush border. But what we're talking about here is the structure of the villus, the position of the apical surfaces of the enterocytes out here, and the basilar surfaces facing the inside of the villus. And this blood vessel here is going to be a branch of the hepatic portal system. So we have the hepatic portal blood is going to flow through this region right beneath the enterocytes. That means we have strategic location where the amino acids and glucose that have passed through the enterocytes can enter this extracellular space, get into the hepatic portal blood, and be swept off to the liver. The next structure in here is in green, and I want to show this here because this is what they call a lacteal. The lacteal is an extension of the lymphatic system. And where the water-soluble nutrients, mostly glucose and amino acids, will be taken up by the hepatic portal system, we'll see that lipids in the form of lipids in the form of triglycerides will now, lipids in the form of chylomicrons. Lipids in the form of chylomicrons that were formed in the enterocytes will enter the lacteal and be swept off in the lymph. Turns out that these chylomicrons are just too big to get between the narrow gaps that are found between the endothelial cells of the capillaries. So we walk around that problem by using the lymphatic system. But let's start here with where we were last time and finish with the idea of the proteins. And first I'm going to draw an individual enterocyte. So this is an individual enterocyte. Remember, the apical membrane is thrown into these folds called microvilli. And this is now going basolateral. This is going to be apical. Before we left for break, the emphasis was on the disaccharidases. But now we're going to put another kind of transmembrane protein out here. And I'm going to label this as an amino and its cousin in red, carboxypeptidase. As the name implies, it converts peptides to single amino acids. And then, in orange, out here in this apical membrane, there's this protein called the sodium-driven amino acid cotransporter. So we can now take individual amino acids, which have been clipped from either the amino or carboxy termini of peptides, take those amino acids and using the sodium-driven cotransporter move sodium, but more importantly, amino acids, into the cell. Then on the basolateral surface down here, just as we saw in the proximal convoluted tubule, are facilitated transporters for amino acids. So the amino acids now can basically, just by diffusion, diffuse out across the basolateral surface. Now remember from the picture we just showed that right next door to the basolateral surface are these capillaries of the hepatic portal system. So just as we saw on the Wednesday before break, where we could move hexosugars, whether it's glucose or galactose or fructose, across the apical membrane, could now move them out across the basolateral membrane by facilitated transporters and put them in the blood. Now then, the question is, what's the fate of these molecules? Where are they going? And we can say here that the hepatic portal blood is destined for the liver. But it's not just being dumped into the liver. There's a very careful structure within the liver which allows us to take up these amino acids and glucose. And this is something known as the liver lobule. So we said that this is the basic structure of the fundamental unit of the kidney. And it's unusual in that it's hexagonally shaped. And that each of these corners, there are going to be 3 tubes. One tube I'll show in blue. This tube is an extension of the hepatic portal vein. The other tube I'll show in red and this is said to be the hepatic artery. Now again, the hepatic portal vein, because all this blood has passed through the gut, in the hepatic portal vein, we find that the blood is O2 poor and nutrient rich. On the other hand, the hepatic artery blood is oxygen rich. The hepatic artery is actually a branch comes off of the descending aorta, so it's oxygen-rich blood. But we see this at each of the 3 corners, each of the 6 corners, rather. Then at each of these corners, the blood joins in a structure. They open up into a structure called the sinusoid. Now, if we look at the sinusoid is lined, as are all capillaries, by endothelial cells. So we have a lining, and I won't draw them all in, but we have the lining here. And from each corner, we have a sinusoid. It comes down. And empties into something known as a central vein. So this is the central vein. So this central vein of the liver lobule is going to drain the blood out of the liver, but let's follow the course. Two forms of blood are coming into the liver. One, oxygen-rich through the hepatic artery. The other that's coming in is oxygen-poor, but nutrient-rich, through the hepatic portal vein. This blood then mixes in the sinusoids. And then right next door to the endothelial cells of the sinusoids are these thousands and thousands of cells. And these cells are known as hepatocytes. These are basically liver cells. Now I'm going to take this pattern and try to reproduce it in the upper board. And what we're going to do is look at the interface between the endothelial cells of the sinusoid and the hepatocytes. And imagine what's going on there. So if this is the sinusoid capillary, then right next door will be the hepatocytes. So these are meant to be hepatocytes. One of the major proteins, one of the major transmembrane proteins in these hepatocytes is one we've seen before. We saw it as a facilitated transporter in the kidney. And this facilitated transporter is GLUT2. GLUT2 is sometimes known as the bulk glucose transporter. And so what it allows to happen is the glucose, which is in the hepatic portal system, which is now in the sinusoid, the glucose can now leave the sinusoid and go into the hepatocytes. It can go into the hepatocytes. It has a pathway. It has an avenue into the hepatocytes through GLUT2, facilitated transporter. And once inside the hepatocytes, glucose is assembled into glycogen. Remember that glycogen is a polymer of glucose. And it's a highly branched polymer of glucose. It's not like amylose, which is straight chain. It's highly branched. So we can now take lots and lots of glucose and store it in these liver cells as this highly branched structure, the glycogen. So we're now going to store glucose as glycogen. But similarly, we have in the nutrient-rich blood coming from the hepatic portal system, we have amino acids. And again, we're going to have facilitated transporters on these hepatocytes. And amino acids now can move from the blood vascular compartment into the hepatocytes. And once in the hepatocytes polymerize amino acids into protein, of course. Most of it is albumin. So again, it's this pattern of taking these great big molecules, these great big polymers, breaking them down to the component parts and then reassembling them. And so we're reassembling amino acids into proteins in the hepatocytes, and we're reassembling glucose into glycogen. But there's a third character out here. Remember I said there were going to be 3 tubes. The third tube, I will put this, the third tube out here I'll put in blue. No, I don't want to do that. I got green. No, I don't want to do that either. The third tube here is going to be part of the bile duct system, because there are a separate set of tubes running next door to the hepatocytes. These tubes, I'm just putting in here as green, these are called canaliculi. And they accept bile salts and bile acids synthesized by hepatocytes. The bile salts and bile acids have this interesting structure. They're based on cholesterol. So there's this 6-membered ring, another 6-membered ring, another 6-membered ring up here. And then out here on the there's a 5-membered ring. And out on this region out here, we can put glycine or taurine. You may recognize that this portion of the molecule is in fact composed of the A, B, C, and D rings of cholesterol. So this cholesterol is hydrophobic. This region out here, glycine and taurine, is hydrophilic. So now, taken together, we have a molecule that's amphipathic. And it can serve as a detergent to emulsify fats. This is the beginning of the discussion of why fats are so difficult to handle. It's pretty easy to digest molecules based on glucose or molecules based on amino acids because they're all water soluble. But remember, triglycerides are hydrophobic, so they're not water soluble. And so what will happen is we'll take these great big globs of fat, we'll call it a fat globule, and we'll use the bile acids and bile salts to convert that, as a detergent would, from this large fat globule to these sort of BB-like structures, which we call fat droplets. Why it's important to do this is that the enzyme that we're going to employ here, the enzyme that we're going to employ to break down the lipids, is one called pancreatic lipase. You may recall that what pancreatic lipase does is it converts triglyceride to a monoglyceride and 2 fatty acid molecules. So it basically takes the triglyceride with the 3 carbons linked by those ester bonds And it basically cuts the ester bonds in two places. By cutting the ester bonds in two places, we end up freeing a fatty acid and freeing a fatty acid down here and leaving behind a monoglyceride. So we're generating out here something known as micelles. We're generating these micelles that are composed of, just number it, they're composed of monoglycerides, they're composed of fatty acids, and they're all stabilized by bile. But we can come back to this upper panel. And again, I'll remind you that out here on the apical surface, out here is the so-called unstirred layer. And the micelles can now, after they've been formed in the bulk phase, the micelles come into the unstirred layer. So these are micelles. Remember, this is a quiet zone. And so the micelles now can bump up against the brush border. They can bump up against the microvilli. And when they do that, the fatty acid and glycerides that were in the micelles can now come right across the membrane. They're hydrophobic, they're lipophilic, they come across the membrane, and they're now in the cytoplasm. So in this way, we've taken lipid in the diet, we've emulsified it into tiny droplets, we've attacked the tiny droplets with the pancreatic lipase, and converted those molecules into fatty acids and monoglycerides. Once we stabilize that as a micelle, then it can enter the unstirred layer, and those molecules can go right into the enterocyte. But then the question is what happens there, and that's where we'll go next. So inside, and come back over here. Let me get rid of this. So what we're going to do now is imagine what's going on with these lipids in the enterocytes. And we'll see that we go through this Rube Goldberg kind of device where we reassemble those monoglycerides and fatty acids to triglycerides. And we're still not done, because once we've reassembled them, then we have to mix them with protein and form structures called chylomicrons. But let's imagine this is one of those enterocytes. Here's the brush border, microvilli of the brush border. There's going to be a nucleus in this cell. And as we saw early on, that nucleus is going to be associated with the endoplasmic reticulum. So this is meant to be endoplasmic reticulum. Now, the endoplasmic reticulum has a particular job here. It's different than the endoplasmic reticulum functions we've seen earlier. And what the endoplasmic reticulum is going to do is it's going to reassemble fatty acids and glycerides into triglycerides. So those things that we tore down with the lipase, we're now reassembling. We reassemble them in the intracellular compartment of the endoplasmic. And then vesicles will bud off from the ER, and they will then join structure we've talked about before. This is, again, fundamental cell biology, where these vesicles now join the Golgi apparatus. And in the apparatus, the triglycerides are complexed. They're going to be complexed with protein. And by complexing, by mixing the protein with the fatty acids, we make them a little bit more hydrophilic. And then budding off of the Golgi stacks will be these structures, which are vesicles that contain something called chylomicrons. Now, the chylomicrons are nothing more than triglycerides. Complexed with protein. These are pretty big vesicles, but they, just like we've seen before by exocytosis, they will fuse to the basolateral surface of the membrane, and they will release these chylomicrons. Now, the chylomicrons are so big that we can't get them into the capillaries. We can't get them into the blood capillaries. So instead, the chylomicrons, go to the right-hand side, the chylomicrons are now going to enter the lymph vessel called the lacteal. Chylomicrons enter the lymphatic vessel, in the villus, which has a name called a lacteal. Those chylomicrons are then carried in the lymph and dumped into the subclavian vein, like all the lymph. So it's just a lymph story rather than a blood story. Now, downstream of this, we do a whole bunch of different things here with other enzymes. We tear the lipid down again and put it back into cells. All of this to show that because lipids are so difficult to handle, but because they contain such an enormous amount of caloric energy, each of those acetates can be put into the Krebs cycle, that we go to great lengths to digest lipid, even though it's a real pain. And we have to go through all these processes because it's such a hydrophobic molecule. So are there any questions up to there? Yes?
So when in the Golgi apparatus, when the triglycerides are complex with protein, does that make them more hydrophobic?
That makes them more hydrophilic. So you're not just dealing with a totally hydrophobic molecule, and that's one of the reasons why you can have them in the lymph. Yes?
How do the micelles get back to the unstirred layer?
It's a one-way trip for micelles. So the micelles are formed in the bulk digestion from the lipid droplets. And the combination of the lipid, so the pancreatic lipase digests the lipids into monoglyceride and fatty acids. Then those are sort of held together as a sort of micelle with the bile salts and bile acids. But then once they get into the stirred layer and they contact the plasma membranes, the enterocytes, then the fatty acids and the monoglycerides, because they're so lipophilic, go right into the cell. And then once in the cell, they're reassembled into triglycerides in the endoplasmic reticulum. Those vesicles then carrying triglycerides in small amounts go to proteins and then form the chylomicrons from that, package those into vesicles, and the chylomicrons then leave the enterocyte through exocytosis on the basolateral side. Does that sort it out for you?
Yeah.
Okay. Anything else? Let me show you a couple of pictures, and then I want to finish the story with the endocrine pancreas. So this is I like this figure because it's showing in 3 dimensions the structure of the liver lobule. And if you look carefully, you'll see that there's this hexagonal pattern, and it shows at each vertex the extension of the hepatic portal vein and extension of the hepatic artery, and also this green structure, which is part of the bile system. Now, the bile is being produced by the hepatocytes and draining into this biliary canal. But then these hepatocytes have the other job of reassembling amino acids into protein and glucose into glycogen. And that's when the amino acids and glucose are taken up from the sinusoids. These purple structures are meant to be sinusoids, and taken up out of the sinusoids and reassembled by the hepatocytes. This is just a higher magnification view showing the mixing of the hepatic portal blood and the blood from the hepatic artery, and then how these structures, the sinusoids, are lined by the hepatocytes. And this structure here now is gathering bile and putting them into these canaliculi and shunting them now off to the bile ducts. And I want to show this picture too. Oh, this one. Then that bile ends up being stored in this structure, the gallbladder. The gallbladder is just a sac. And you remember the term cholecystokinin, bile, sac, move. Well, cholecystokinin binds to smooth muscle in the gallbladder and causes it to contract and force bile out into the small intestine. But it also, remember, has that other effect on the acinar cells. This other figure is one I'll never ask you to reproduce, but it's useful to think about, and then it's a more detailed picture of what I showed on the board. This is cholesterol. This is glycine out here linked through this structure on this carbon in the pentagon. And up here again, this is now going to be taurine out here. And these structures are hydrophilic. They're charged. Remember, or at least this one has a charge on it. This one has a double bonded oxygen, so it's polar. And now this is the polar end of the molecule. This is the hydrophobic end. It's amphipathic, and it functions as a detergent. The last few minutes, let me finish with this story. Of the endocrine pancreas. And I want to tell this story because it's so important to so many of us. We all know someone who is diabetic. And I'm going to simplify the story and talk about the pancreas. And you remember that our discussion of the pancreas was this sort of crenellated structure of the exocrine pancreas. And it emptied into the common bile duct. But then outside of this exocrine pancreas is additional tissue. So this is the exocrine pancreas. But outside of that, there are clusters of cells. These are the beta cells of regions called the islets of Langerhans. Now, these beta cells, the beta cells also contain GLUT2. And in response to high blood glucose, So after a meal high in glucose, that blood will, not all of that glucose is going to be taken up by the hepatocytes. And it's going to spill out into the central vein and be taken off back into the venous system and pumped through the cardiovascular system. Well, when it comes back to the liver, these beta cells recognize high glucose because they take up glucose through GLUT2. But when they take up glucose through GLUT2, the beta cells secrete a hormone called insulin. And if we look at muscle cells, whether they're cardiac muscle cells or skeletal muscle cells, on the surface of these cells are these transmembrane proteins called insulin receptors. The insulin receptors will bind insulin. So the insulin has been released. The insulin has been released by the beta cells. It binds to these receptors, and in response, vesicles fuse the plasma membrane. And these vesicles contain a glucose transporter called GLUT4. This glucose transporter allows glucose to go into these cells. Without GLUT4, glucose doesn't have access to these cells, or at least not very easily. So the function of the insulin is to allow glucose into the cells. To finish this quickly, what happens in diabetes is two things. In most cases, the problem is that we lose function of the insulin receptor. And that's type 2 diabetes. But there's another form of type 1 diabetes that's even more devastating in that we kill the beta cells. And then we're constantly needing, for the rest of our lives, we need to inject insulin. And we're now, it was just an article yesterday, they have generated, we'll see if it works, they've generated some stem cells that act like beta cells, and they've put them back into this diabetic person's body, and that person is now responding and doesn't have to take insulin. And so there's this faint hope, we'll see how it turns out, there's this faint hope that stem cells can be used to rescue type 1 diabetics. And with that, I'll let you go. That's my last lecture, and so I